Okay, so I feel a little bit uncomfortable here because I'm not a theoretician. This is not a theoretical talk. Not only is it an empirical talk, it's an empirical talk without a ton of data. So I'm talking about a brand new project on something that's a little bit outside my wheelhouse. So why did I decide to talk about it? I think that microchimerism offers a lot of intriguing questions about the possibility of internal conflict. There is a tremendous dearth of data on this topic that I'm trying to correct. I also think there's a dearth of evolutionary theory on the topic, but I will acknowledge that of the theory that's been done, almost everybody's in this room. So it seemed like that was a reason to try to talk about this in the state that it is. Okay, so as we've talked about before, many multicellular organisms originate from a single cell that divides to form most of the cells of the body. And so this high level of clonality or relatedness is thought to be really important in limiting cell-cell competition and reducing the opportunities for conflicts within multicellular organisms. I spent most of my career working on an organism that does not start from a single cell, that undergoes what we call chimeric development. So cells aggregate to become multicellular. This means you can have multiple genotypes within the multicellular organism. And in this case, in these chimeras, as we said, different agents, different agenda, they may want to push phenotypes in different directions. The cells can be at cross purposes. So in general, as evolutionary biologists, we look at chimerism as potentially a hotbed for conflict. But this distinction between clonal versus chimeric organisms is not cut and dry. It varies along a continuum, and we can talk about a phenomenon called microchimerism, which refers to very small numbers of non-self cells present in an individual. And in fact, everybody in this room is probably microchimeric. So we know that during pregnancy, cells pass between the fetus and the mother. So you may have cells of your mother throughout your body. And if you've given birth, you may have the cells of your children dispersed throughout your body, including in your brain. Microchimerism is one of those things where it's thought to be a ubiquitous outcome of development. And so I would say people, when they go to look for it in placental mammals, tend to find it, but there's still not that much data other than in, I would say, humans and mouse is where most of our sort of quantitative data about this phenomenon occurs. So just a little bit of example about this. So in the journal Molecular and Human Reproduction, you had some researchers that studied through autopsy women who died while pregnant with male sons or within a month of giving birth. And this is looking at how many cells they could find that contained Y nuclei, so presumably of fetal origin, in different tissues in the body. And so we're saying about out of 10 to the 6 cells, they had about 10 to the 2 of these fetal cells dispersed throughout their body. So it's known, at least from humans, that this peaks during pregnancy and declines very quickly after birth, but it does not go to zero. And so in this kind of cool study from Lee Nelson's group, they examined the brains of deceased older women, and they found Y chromosomes present in 63% of them. And in some cases, these are brains where the woman was not known to have given birth to a male son. So I've talked about microchimerism arising in utero. It's also well known to occur among twins through transplantation and also through transfusion. And you may or may not know that it actually also occurs through nursing. So mothers transmit immune cells to their offspring, and it's been shown that that enhances the development of the baby's immune system. So babies will show tolerance to non-inherited maternal antigens they were presumably exposed to through this process. And if you talk to immunologists, they will say that this is why they do it. So mothers introduce these cells so that they can train their baby's immune system. And if you're an evolutionary biologist, you might have other questions about what these cells might be doing. So some major problems in the field. There have been major methodological limitations. You're looking for these extremely traced cells. You're often looking at small numbers of samples, opportunistically collected, and a wide variety of techniques where you're trying to detect something at a very low frequency that vary in their technical variability. So it's very hard to compare these across different studies and to make sense of the numbers that they're getting out, as well as the fact that the studies that really do any kind of quantification of this are pretty much humans and mouse. And so we have, with these methodological limitations and so few species in which this has been studied, we can't really even begin to address the evolutionary questions of what are these cells doing and can they possibly mediate a conflict, particularly parent-offspring conflict within organisms. And so these questions have been out there for many decades now, but I don't think that there's been much progress in being able to address them. So I decided I wanted to work on this problem. And so one thing that I'm hoping is that some new technological advances that have not been really taken advantage of in the microchimerism field yet offer some hope for quantifying microchimerism cheaply in such a way that we could study it in depth in one species or across a broad range of species. I can't say yet if I can do better. I think I can at least do as well in terms of sensitivity. So here I'm just showing a picture depicting maybe what you would see in a tube of DNA from a mother and an offspring. And I'm just going to organize these DNA strands a bit so that we can see them. And you can see that in the mother, we expect half of her DNA to have her paternal haplotype. The other half should have the mom's maternal haplotype. And in the offspring, we can see that the offspring inherits its mother's alleles, but it's going to be recombinant, a different haplotype. It's going to inherit its father's alleles, which I'm showing in blue and yellow, but it will be recombinant for those. And you can see in here at very low levels, you expect to see, if there's microchimerism, small numbers of molecules that are a match to a different individual in the population, probably a first-degree relative, if this is true microchimerism. So this is what I want to try to detect through sequencing two major haplotypes. I want to fingerprint the whole population, and then I want to detect these rare haplotype sequences. I think long-read sequencing, so in part because you can get high-depth single reads easily, but also the fact that you can get very, very long sequences. So let's say up to 50 kb on a single strand means that you're getting that sophisticated haplotype information. So it's not just that you see some SNPs that match another individual, but that you see, say, 50 SNPs in a row that are a perfect match to the offspring's mother. So that's where I think the power comes in. So other weird thing, I decided to study this in spotted hyena. So why spotted hyenas? First of all, I just want to point out Kay Holkamp, who's the founder of a 40-year project in the Masai Mara in Kenya, studying one main clan and some subsidiary clans of spotted hyena. So for 40 years, she's just now retiring. And so Zach Loback is also working on this project. He's the new co-director of the project. So why spotted hyena? So the first thing is that they have a hemochorial placenta. And so what this basically means is it has the most invasive type of placenta. It's a type of a placenta that we see in primates, we see in humans, we see in primates, we see in rodents, we see in rabbits, but it's been lost throughout most of placental mammals and then was actually re-evolved specifically in the hyenas. And I think that reflects some of the need that offspring have to grow big fast in utero. There's also, I think, a lot of parent-offspring conflict is particularly strong in this system. So hyenas have an unusually long and lengthy gestation given their body size. They nurse for extremely long periods of time, 12 to 18 months, and they have the most nutritionally rich milk of any carnivore. So there's a major investment that a mother is making in her offspring. The other interesting thing is that there's extreme levels of sibling conflict. So they often have twin litters. So that's another opportunity for there to be, they often have twin litters. So another opportunity for microchimeric cell exchange. They are born with their eyes open and their teeth erupted, and they've been observed attacking their litter mate within hours of birth. And so why do they do this? It's most likely because they have a rank, so they have inheritance of rank in terms of priority of access to kills. So for the most part, they're going to slot in beneath their mother's rank in terms of getting food, with the exception of these twins. When they're born, they need to fight and establish a lifelong rank differential that will determine which of them feeds first at a kill. So they often kill each other. The other interesting thing is the new cubs are going to slot in above an older cub in the ranking. So older offspring will have a major interest in preventing the birth of their younger siblings because they're about to get outranked by them. And then the other reason is because the Mara Hyena Project has an incredibly rich source of data. So birth to death demographic records covering 40 years. So we know everything about how much they've reproduced, records on how much they've been nursing, sometimes even the amount of nursing transfer, multi-generational quantitative data, but most especially biobank samples. So there's over 900 DNA samples from these individuals, from 581 hyenas, and more than 100 parent-offspring pairs, where I also have access to twin data. So if there were any place to look for it where I think individuals are, where there's likely to microchimerism can occur because of the type of placenta, the twin births, as well as the incentives there for conflict between parents and offspring and between siblings, I feel like this would be the place to look. So if you're just starting with this and you've got these very precious DNA samples, I have just 11 samples that I started with. These are males or females, different conditions, so lactating, pregnant and lactating, and some that have never given birth, and they vary in how many male offspring they've produced. 2 individuals, Coco and Rs, this is a mother and a cub, a male cub. As a starting place, we went through and sequenced the whole genome using long-read sequencing to get very, very high-quality reference genomes for these 2 individuals. This is so I have some ground truth when I start trying to apply these tests to detect microchimerism. Eventually, I haven't done it yet, but I'm planning to simulate microchimerism in part by swapping reads between the different genomes and then at different levels, and then seeing how well I can actually detect that. So if you don't know what you're doing, and this was before I had any funding for it, what's the first place that you would start looking for microchimerism in these organisms? So I started with trying to amplify a Y chromosome-based locus in females. So this should not amplify unless they've gotten some male cells. So I did a nested PCR. So these are outer primers. You amplify, then you use the PCR product from the first PCR for a second PCR. And what this ends up being is an extremely sensitive test to pull out extremely rare SRY alleles in a DNA sample. I'm sorry, this is such an ugly picture, but the upshot is on the left, I have my first round, 2 examples. I've done this a million times. 2 examples of first round PCR. Males and only males produce SRY products there. But when you take that PCR product and you do the inner one, then you start to see females producing products, suggesting that they have low amounts of SRY DNA present in their DNA samples. What got really weird, and I tore my hair out over this for quite a while, is that these are the same individuals across independent PCRs, and it was idiosyncratic. So sometimes the female would produce a band, and sometimes she wouldn't. And this bothered me a lot until I started thinking about what I was actually doing and calculating how many molecules were probably in my original DNA sample. And then you're putting it through an exponential process where somewhere in there, the polymerase is finally going to find that rare template and bring it up exponentially. And so I think you're going to tend to get this weird idiosyncrasy just depending on when that template happens to get picked up. The good news is you can do better. So I was always going to move to this, but I did the regular PCRs first. I do something called droplet digital PCR, which works pretty well. So here you're taking a PCR reaction, and instead of doing it in one well-mixed tube, you're actually distributing your PCR reaction into lots of little nano droplets, about 10,000s of them. And you're distributing it in such a way that most droplets don't receive any template. They have no PCR reaction, and then others receive template according to the Poisson distribution. And when the PCR is successful within one of those little nano drops, it will turn a color. So you end up with this digital output of positive negatives across numbers of tens of thousands of microdroplets. This is just a trial run with some technical replication, but not true replication, just showing that I have my negative control. This is on the y-axis, the concentration of the SRY, an estimate of the SRY concentration in copies per microliter. Negative control looks good. My 2 males up here have really high concentrations of SRY. one of my males failed. And then I have a whole bunch of females where at least so far they're barely eking out above the negative control, but they're there and it's one time. So my hope is that by the time I add some optimization with probes, with replication, that I may have a decent quantitative measure to say these individuals vary in my estimate of how much microchimerism they have, which is what I'm looking for. I'm going to do better than this. So SRY is only going to tell me about male microchimerism. So I'm ignoring then all of the examples of microchimerism introduced by female births. And so what I wanted to do was to come out with a better way of identifying genetically an exact individual. And so I took my genome assemblies, high quality. They managed to pull out a big chunk of the MHC locus in one go, which is actually saying a lot about how good these assemblies are these days with long reads. Took the 5 haplotypes for which I have any genetic information and align them. And the upshot is I can find regions of the genome that are extremely relatedness informative, where I've got the most SNP information to distinguish different hyenas. And I've started designing primers so that I can amplify just in those regions, get the high copy numbers I need to have a sensitive test of microchimerism. It's not trivial. Actually, that's still underway. So project goals for the hyena study is if I can come out with this nice quantitative measure of microchimerism in the hyenas, I have this huge data set for 40 years to associate with it. So I can ask questions like, are microchimerism levels affected by parity? So how many offspring they've had? Or is it less about access to that tissue and more about the ability to remove those cells actively later? So maybe it's going to be related to genetic differences at immune loci, or maybe most of this microchimerism comes about through nursing, in which case I may see relationships between microchimerism and the amount of time that they've been nursing. And again, all of these records are present. As I've gone through and done this work on the hyena, though, I became slowly convinced that the real evolutionary questions would never be really answered in one species, but that you really have to look across species and that it's a comparative study that we would benefit the most from. So I'm hoping if I set up these techniques in hyena for how you do this, that I can export that to any other species that I want to look at using the same technique. So then we can start saying, how does microchimerism levels vary among placental mammals? Is it true, as is cited without any evidence, that it's actually driven by the different types of placental morphology and how good of a barrier they are? Again, could it just be that it doesn't matter what the initial barrier is, it matters how well you can clear them out, as well as other potential explanatory variables? And so to that end, I'll just say I'm about to go back to New Zealand in a few weeks, and I'm starting now to try my hand at marine mammals. And so my former colleague, Karen Stockin at Massey University, has one of the largest tissue collections in the Southern Hemisphere for beached whales, basically. So she's the person who gets called when you have mass strandings. She sent me these pictures, which will probably be the first cetaceans that I try. This is a female dolphin who died while pregnant, which is apparently very rare, with twins. And so she has saved all of the tissues for me, placenta, umbilicus, the twins, the mother. So I have a lot of opportunity, and there's a good genome for it, to try this out and start branching out across the evolutionary tree. Okay, so just some thank yous. So the Mara Hyena Project has been amazing. Kay Holkamp and Zach Loback. On the genomics side, collaborators at the Liggins Institute at the University of Auckland and at the University of Washington and Seattle Children's, Danny Miller. These are all people who are major experts on long read sequencing and genome assembly. And the Hypothesis Fund for funding this when it's just a hypothesis, and the UW Research Royalty Fund.
I think we have time for maybe one question while we get it set up with the next speaker.
Should I grab the mic or? Okay, thank you.
Thank you. Great talk. A very, very, insightful, intriguing project. Good luck with everything. I was just curious on the methodological side of things. how readily do you get high molecular weight DNA from those samples, particularly if they've been stored for 40 years for long sequencing? Thank you.
Yeah, so we did fine. So with Coco and ours, I just took the DNA that I had readily available, and we did great with that. I mean, our reference assembly with 150X is as good as the current reference that we have. I am assuming that many of these samples will not be good quality, and that's where the droplet digital PCR can help. It needs trace amounts of DNA. And with the MHC, I'm looking for regions that are around 10 to 15 kb. I may pool multiple loci together, but hopefully I can still amplify. That's still going to be a bit tough, but amplify a bunch of different regions that are relatedness informative. But by reducing the fraction of the genome that I'm looking at, I need less DNA, and I'm getting the really high coverage that I'm going to have to get to be able to pull out these rare haplotypes. So it'll be varied. Some things I'll do more genomics on, and other things are going to be more PCR based.
Okay, thanks.